Hi everyone, my name is Sunkyu Kim, a graduate student in Korean University. First of all, I'm really proud that I can give a talk in the best conference of bioinformatics. Thank you for the committees in this conference. Today, what I want to talk about is improved survival analysis by learning shared genomic information from pan-cancer data. Okay, the survivor is one of the most important information for patients with severe illness and their families. It is totally different with the cell death or drug perturbation kind of death in a wet lab. The survivor is one of the most important and ultimate phenotype. So, lots of clinicians and researchers have tried to predict the survival time or the risk of death of patients. Recently, with the explosion of genomics data of cancer patients from several consortiums, including TCGA, ICGC, kind of that, many researchers have applied machine learning techniques to predict the survival of cancer patients using gene expression data. One of the most important and most famous survival model is Cox proportional hazard model, which was introduced in 1970s. The basic assumption of the Cox model is the probability of survival over time follow an exponential function, and the slope of each survival function, survival curve, is determined by the patient feature. And the patient features, like expression of genes, are aggregated by weighted sum and represented as single value. But the linear combination of gene expression is too simple calculation to capture complex signals of genes. So in the last year, there was a new survival model that used the deep learning technique to aggregate the patient gene expression values. However, when applying complex models like deep learning, there's always overfitting problem. We all know that the number of genes is more than 20,000, but the number of cancer patient samples is not that many. So even though the breast cancer has the most samples among cancer types provided in TCGA, but the number of samples of breast cancer is just around 1,000. And the overfitting problem causes a model to show poor performance at the unseen samples, including real situation. So, one of the representative solutions of overfitting problem is transfer learning. Transfer learning is a model training technique that extracts a certain knowledge from a trained model and transfer the knowledge to a new model to be trained. In this task, the transferred knowledge, I assumed, is a shared information from gene expressions of heterogeneous cancer type samples. First, I trained the variational autoencoder model using gene expression data of TCGA pan-cancer patients. Then, I transferred the knowledge, the shared information of pan-cancer learned from the pan-cancer gene expression data to the survival prediction model for each cancer type. Finally, I tried to interpret the survival prediction model to check out it really predicts survival based on biology, not just statistics. Our model name is VA Cox because it is combination of variational autoencoder and the Cox proportional hazard model. The main objective of the pre-training phase is extracting common cancer knowledge of multiple heterogeneous cancer types. We considered using simple autoencoder, but the simple autoencoder is prone to overfit on some features among various cancer characteristics because it aims to perfectly reconstruct the given input data. So we selected the variational autoencoder for pre-training. Variation autoencoder uses the objective function based on the reconstruction loss between the given input and the decoded output based on the randomly sampled vector. Since the variation autoencoder uses a random-based loss, it can learn robust features among various cancer characteristics. Okay, then the survival prediction phase. The main objective of a survival prediction phase is prediction of deterministic patient survival, not generating probabilistic samples. So we transfer only mu encoder weights and combine Cox proportional hazard layer that calculates the hazard ratio. Right? This is the result part. We used Harris concordance index as a primary metric to evaluate our model. And we split the dataset into training and test set 10 times and obtained 10 concordance index scores. And all the hyperparameters, like a number of hidden nodes or regularization kind of that, are tuned based on 5-fold cross-validation over training set. We find that our VA Cox outperformed other baseline models at 7 cancer types among 10 cancer types. And our VA Cox outperformed other baseline models in terms of the average concordance index of 10 cancer types. We also drew the Kaplan-Meier plot and calculated the p-value using Logrank test. To do this, we split the test set into high and low risk groups using predicted risk values of samples. And the result is our VA Cox also outperformed other baseline models in most cancer types. Especially at BLCA, the bladder cancer, our model did not outperform baseline models in terms of concordance index, but in terms of Kaplan-Meier plot and Logrank test, our VA Cox model significantly outperformed the baseline model. Okay, alright, the remaining 2 cancer types where our VA Cox did not perform the baseline models are the LUAD, lung adenocarcinoma, and the LUSC, lung squamous cell carcinoma. Both cancer types are the subtypes of non-small cell lung cancer. And we all know that, and the non-small cell lung cancer is well known that has very heterogeneous and distinct genomic characteristics. So we can find the transferred knowledge about common cancer characteristics of gene expression gives rather negative effect to the survival prediction model on the 2 cancer types. We also performed ablation tests. First, we compared the pre-training models, simple autoencoders, denoising autoencoder, and the variation autoencoder. Among them, the variation autoencoder shows the best performance when used to extract the common cancer knowledge. And the poor performance of a simple autoencoder shows our intuition about the random-based loss is right. We also check whether the transfer learning is effective. We tried possible options about transfer learning and find out our current option shows the best performance, which is noted as red colors. One interesting finding is the comparison between no weight transfer and weight transfer with the fine-tuning, which is our current option. The model of the weight transfer with the fine-tuning outperforms the model of the no weight transfer option at exactly same 7 cancer types where the VA Cox outperforms baseline model. It means because the baseline models did not use transfer learning, This result demonstrates that transfer learning is effective for survival prediction. Okay, this is the model interpretation. I'd like to find what signals the hidden node captures to predict survival. So I considered the expression values for each gene over patients as vectors, and hidden values for each hidden node as also vectors. And then I calculated the Pearson correlation between the hidden node vectors and the gene vectors. This is the visualization of the correlation table. You can find that the most correlated genes are cancer-associated genes. First, the CDC20, C9orf86 known as RBL1, and the MAMDC are known as survival-associated genes. Among them, especially CDC20 is a very well-known survival-associated gene for breast cancer patient survival. And other genes, including MAMDC2, HJURP, kind of that, are also known as cancer-associated genes. Based on the correlation table results, you can rank the genes for each node using correlation values. Using the ranked gene list for each hidden node, I did pathway enrichment analysis based on CAG pathway database. The colored box means the pathway has statistical significance, the p-value lower than 0.05. Then I find the cancer-related genes and cancer-related pathways for the hidden nodes of the last layer. Then, what about the second layer? I did exactly same analysis for the hidden node of the second layer, and I found the metabolism-related pathways, including fatty acid, propanoate, retinol metabolism. I think it's very interesting results, because we all know that the central dogma in molecular biology, the information, the DNA information, including mutation and copy numbers, affects RNA transcriptions, and the number of RNA transcripts are correlated to the protein abundance. The proteins interact with each other and generate metabolites. Then finally, the phenotype is observed, right? Based on the interpretation analysis above, our model shows it somewhat follows the central dogma, right? Our model predicts survival based on the central dogma of molecular biology, because the model inputs gene expression values, which is transcriptomic information. The first hidden layer captures complex signals from input the gene expression information, right? And then the second hidden layer captures more complex signals based on the first layer's signals. And we already find the second layer captures metabolomics related signals, right? Then the third hidden layer also captures much more complex signals based on the second layer signals, right? Then we already find the third hidden layer captures cancer-related function. And finally, based on the third layer signal, the model predicts survival of a patient, which is the ultimate phenotype. Right? It's very interesting. Actually, we have tried to investigate the first hidden layer, but there is no pathways for each protein translation, so we'll leave it for the future work. Alright, in this work, we propose a deep learning-based survival prediction model using transfer learning. We show the shared information among heterogeneous cancer types is beneficial on survival prediction. And we can find Our survival prediction model follows the central dogma, which means the model predicts survival based on biology, not just statistics. And in the future work, I'd like to apply biological networks like the pathways or protein-protein interactions for better representation of gene expression data. Because the genes are not independent but associated each other, so the pathway or protein-protein interaction network will be beneficial to survival prediction as a prior knowledge. The other future work is multi-omics integration. TCGA provides other omics information, including mutation, methylation, or even protein abundance, like that. So I think it will be also beneficial to the survival prediction of cancer patients. Okay, this is the end of my presentation. Our group have won many challenges, including dream challenges and bio ASQ. And the research topics are bioinformatics, computational drug design, and biomedical natural processing. Alright, thank you for attention.